I'll just get started introducing our first speaker today. That's Mathias Nielsen. And when you look at Matthias's CV, it seems like he figured out really early that he wanted to work on chromatin and non-coding RNA in plants, because this is already what he studied during his bachelor research project at the University of Copenhagen. But then also, as he was working on his master's degree still at Copenhagen, he did a project in the Marquardt lab on the repression of intragenic transcription start sites. And that was a really cool project. And actually, you can watch back Sebastian Marquardt's talk that was given earlier in the series if you want to hear about that work. But after completing his master's, he moved to Norwich in the UK to join the lab of Carolyn Dean, obviously a very cool lab. And there he's been studying the mechanism of polycomb-mediated epigenetic switching in Arabidopsis. And I'm very excited to hear today what he's learned so far. Please go ahead, Matthias.
Thank you very much. And thank you for letting me present my work here, this amazing seminar series. So as I said, I'm going to talk about how to make PRC2 specific, and more precisely the role of accessory proteins in epigenetic switching at a plant locus. So in our lab, we're interested in how plants sense and remember different environmental clues, particularly how plants sense and remember winter, and uses this clue to time their transition to flowering at the right time of spring. So to understand this at the molecular level, we don't study this in trees, but we use the model plant Arabidopsis thaliana, where certain versions also require long duration of cold to flower at the right time of spring. So the research in our lab and many others has shown that this comes down to one central gene known as FLC, which is short for flowering locus C. And you can think of FLC as the brake on flowering. So when FLC is on in the autumn and is expressed, it prevents the transition from vegetative growth to floral growth. But then as plants enter winter, FLC is slowly and gradually repressed in a process known as vernalization. This repression is then remembered in the following spring, giving rise to flowering. So since this is a memory mechanism, the memory is actually stored in the chromatin. So we can look at the chromatin at FLC. This can roughly be divided into 3 different chromatin stages: an active state where FLC is actively transcribed and the histones are enriched in activating chromatin marks like H3K36 methylation and H3K4. Then as plants goes into cold, FLC is transcriptionally silenced in a process that relies on antisense long non-coding RNAs. Then later on, you have deposition of K27 trimethylation, these repressive chromatin markers, at an internal position around the intron 1 exon 1 boundary at FLC. Then as plants goes back into the warm, this repressive marks is spread out across the locus and FLC is epigenetic and stably silenced, allowing the plants to flower. So the long duration of winter actually allows us to study these phases in detail, independent of the other phases. And in my PhD, I've been interested in this middle phase in winter, where K27 trimethylation accumulates at this internal position. I'm sure many of you will know that K27 trimethylation is the product of the enzymatic activity of PRC2, polycomb repressive complex 2. And similar to K27 trimethylation, PRC2 also accumulates in this internal position, as you can see here by a protein chip of one of the enhanced of cis homologs in plants known as swinger. So you can see here by chip, it also accumulates very precisely in this internal location near the exon 1 intron 1 boundary. So this leads to the question of my PhD of how PRC2 accumulate at this internal position. What does it make PRC2 specific for this? So in more general terms, this is similar to say how PRC2 finds its target sites, and is again similar to the question that is asked in many other polycomb systems of how PRC2 is actually recruited to its target genes. So to try to understand this and solve this question, we put together an interdisciplinary team consisting of molecular plant biologists, you have the John Innes Center, and then structural and biochemistry expert down in Cambridge in the MRC Laboratory of Molecular Biology, with Elsa and Mark based in Marianne Bind's lab. And this was possible really by an ERC grant to the lab. So could take a step back and look at the core player in this process, PRC2. I'm sure many of you will be familiar with the core components of PRC2 from mammalian and flies, with SUSI12 as a scaffolding protein, EED and RBP4 and then enhanced of cyst, 1 or 2 as the enzymatic component. So in Arabidopsis and in plants in general, we have really conserved components of PRC2 core, but we also have an extra layer of complexity in that we have several homologs of some of these core components. Like we have 2 homologs of SUSI12, WERN2 and EMF2, and we have 3 homologs enhanced of cyst, Swinger, curlyleaf, and Medea. So even by changing the core components, we can make PRC2 specific for some of the targets. And here I've highlighted the complex that is involved in vernalization, which is consisting of WERN2 as the SUSI12 protein homolog, and Swinger as the enhanced of cyst. However, recently, a lot of work has been looking into how you make PRC2 specific by changing the proteins that associate with PRC2. So again, to bring it back to the other polycom systems that might be more familiar to you, we talk about 2 main subcomplexes of PRC2, PRC2.1, which is associated with PCLs and EPOP, and PRC2.2, which is associated with JARET2 and ABP2. In contrast to the core components, we don't really see conserved accessory proteins in Arabidopsis when we look at the amino acid level. So we need a different approach to find accessory proteins. And here it turns out that the genetic screen that had been done in our lab and others was available too. So just to give you an example of what had been done, is if you take a plant with a functional PRC2 and a functional candidate protein named WERN5, and you expose it to cold, this plant is now early flowering when you take it out of the cold. However, when you knock out core PRC2, like WERN2, for example, the plants no longer respond to cold and is late flowering. Similarly, when you knock out WERN5, this candidate protein is also late flowering, suggesting that WERN5 works in the same pathways as polycomb. So to try to confirm this, we performed immunoprecipitating mass spec of a YFP tag WERN5. So when we do this in warm conditions, so before the plants have seen any cold, which is the same as a plant in autumn, we see that WERN5 is expressed and we can pull it out, but it doesn't really associate with any PRC2 components. However, if we do the same experiment in the cold, like in winter, we now see that we can pull out the core component of PRC2, MSI1, Phi, Swinger, and WERN2, as well as this protein named Win3. So Win3 is in fact a cold-induced protein, and is therefore likely to think that Win3 is required for the interaction of WERN5 with core PRC2, although we're not completely sure about the mechanism of this. So what is WERN5 and Win3 actually? Well, these are homolog proteins and actually member of a 4 protein family in Arabidopsis, consisting of WEL1, WEL2, and then WERN5 and Win3. And here on the right, you see the domain architecture of these proteins, consisting of a zinc finger, a PHG, a wing helix-like domain, an FN3 domain, then a long linker region, and a WEL domain in the end. So we think, based on this domain architecture, that this would be involved in chromatin targeting. And then we know that the WEL domain is a protein-protein interaction domain. And I will tell you in the end why we think this is actually a polymerization domain. When we first saw this, we were quite intrigued because this domain architecture reminds us of what we see in some of the other accessory proteins from other systems. So here you see MTF2, for example, that also has several chromatin reader domains and a chromatin reader domain and a DNA binding domain, the wing helix domain. So this tells us that even though we don't have sequence conservation of the accessory proteins, we might have a similar mechanism based on this similar domain architecture. This tells us that what we learned from the plant system might be applicable to polycomb system in more general. So to start to understand the function of these accessory proteins, we took an in vitro approach and expressed the different domains in vitro and asked for their function. So we started with the PhD domain, and this is mainly work by Elsa from LMB. So the canonical PhD domains are known to bind histone, tails, and particular H3K4, methylated or unmethylated. So for a long time, we tried to find histone tail ligands for the PhD domain, but we couldn't really find any, and that was quite surprising to us for a long time. Until Elsa recently solved the crystal structure of the PhD domain and showed that the surface charge was positive, which is different from most other canonical PhD domain. And this really suggests to us that it is not involved in K4 tail binding, but more likely has other roles. And one option was that the PhD domain was involved in protein-protein interaction. And indeed, Elsa have recently done a series of co-IP experiments and can show that actually if you take a PhD domain away, Wern5 does no longer interact with the core polycomb complex. Interestingly, another accessory protein from the fly system, PF1, this PhD domain has also been involved in interaction with the core PRC2. So again, this suggests that there could be some core mechanism in the polycomb system between plants and other systems. So moving on to the next domain, the wing helix domain, Jürgen Müller and others has recently shown that the PCL through the wing helix domain can bind DNA and thereby enhance the dwell time of PRC2, leading to K27 trimethylation. So we wanted to add something similar, and to do this, we express the wing helix domain, again in E. coli, but it turns out that you actually need the entire region to get binding to DNA. So we express the zinc finger PhD wing helix and test this on a range of different DNA binding assays. And here you see one example to FLC double-stranded DNA, and you see that this is able to actually bind DNA and cause a shift on EMSA analysis. I should say that this is true both for FLC DNA and also other sequences. So right now we cannot say that the wing helix is specific for FLC DNA. So this is not only true in vitro, but also if you look in vivo by protein chip of Win3, we see that this accumulates also over this internal region at FLC very specifically. So Win3 can also in vivo associate with chromatin and comes down in the same region as PRC2. When we looked at the affinity for this DNA binding, it is quite low. And that then raises the question of how do you actually accumulate PRC2 if you have very low affinity or quite low affinity for chromatin. And here it turns out that our collaboration with Marianne Bin's lab, which has been leading the field in the dishevel signaling world, was a great collaboration, because in signaling, you also often don't want to signal too early and therefore have low affinity for the ligand. And similar here, we don't want plants to flower too early and silence FLC before they have seen the full duration of winter. In signaling, we often overcome this by what is known as the avidity effect, by up-concentrating the protein. And this led our interest into the well domain, the C-terminal domain. So the well domain is a protein-protein interaction domain, and you can see here by co-IP experiment that is completely required for the interaction between Win3 and Wern5. And this is true both for the hetero interaction and the homo interaction of the Win3 and Wern5 to Wern5. If we take the well domain and express it in E. coli and run it over in gel filtration column, this is mainly work by Mark from LMB, you see that it elutes earlier than you would expect for the monomer. So in this system, the well monomer is around 20 kilodalton, but the main peak is around 70 kilodalton, indicative of a polymer formation. Interestingly, we also tested the C-terminal domain of MTF2 and could show that this was actually able to form dimer on the gel filtration column. So again, this suggests that there may be some kind of common mechanism also when it comes to the C-terminal region of these proteins involved in the protein-protein interaction. So this now takes me back to the original question of how PRC2 goes to the nucleation region, this internal region at FLC around the X and 1 internal boundary. So the model that we have in our head in the moment, and this is really a working model, I should say, is that Win3 through the wing helix domain recognizes some kind of feature at this position at FLC. And then you will start through the well domain to build a Win3 polymer scaffold. And this then, in the end, recruits Wern5 again through well-well interaction. And Wern5 through the PhD domain brings in PRC2, which can then add the K27 trimethylation and switch off FLC. So this is one way of showing the model. Another way that we often use to show is this way. So here you can see that we can have several Wern5 PRC2 complexes that docks to a Win3 scaffolding polymer. So this is another way of up-concentrating PRC2 at the nucleation region to facilitate K27 trimethylation. So now we have this very nice model to go from, and we can now start testing this model. And the first thing that we kind of did was to find a way to stop this polymerization to form. So through a range of different assays, we identified 2 amino acids that is crucial for polymerization formation. So here you see a similar concentration. If you take the wild type well domain, it has polymer formation again. the monomer is around 20 kilodalton, but it elutes earlier, suggesting it forms a polymer. But then if you take and make these 2 point mutation in 2 arginines, it's very close together, so likely forming the interface, we see it now runs as a monomer. So now we have a very good candidate to stop polymerization without disrupting the entire protein by making big deletions in the protein. So we can now take these mutations and move into the plant system. So that is what we are doing in the moment. So we make lines where we have deleted these different domains, the zinc finger, PhD ring helix, and so on, but also where we make more soft mutation by blocking the nucleic acid binding interface. And as I just showed you, we now have mutations that also stop the polymerization. So we are really using all our structural and in vitro work to design very clever point mutation in these proteins to test in planta. And this is ongoing work, and we are generating many lines to test this in a moment. And one assay we are using is looking at the microscope, because if you look at wild type Win3 after 4 weeks of cold, you see you get these foci in the nucleus of GFP signal. And this is done by Sylvia in the lab. So prediction would be that if you take the well domain away or make these polymerization blockers, you will have a more homogeneous population signal of GFP. But this is really work ongoing, and we're really testing all these lines in planta in the moment. So hopefully soon we'll be able to tell you about the in-planta effect of these domains and these point mutations that we have identified. And with that, I would like to end and thank everybody in the lab, particularly the one involved in this project, which has been Anna, Mark, and Elsa, and of course Caroline and Marianne, the group leaders. Everybody in the Dean Lab and Martin Hauer's group that we collaborate with, and actually initiated this whole idea of polymerization at the nucleation region. The people I have met and collaborated in both Marianne's group and in Laurie Prasimo's group that helped set up the EMS analysis. Of course, amazing support groups at JIC that help all our plant work has been really great help. And since this is now my last year of PhD, then I would also like to thank everybody at the Joint Center and the Saints Lab for having a fun time here in Norwich. And of course, all the funding that funds the lab, and the BPSRC for funding my PhD. And thank you.
Great. Thank you so much, Mathias. That was a great talk and fantastic work. Okay, I'll just remind people, you can either type your question in the Q&A box, or you can raise your hand, and I'll keep an eye on the participants window. And maybe I'll start off, because at the very start of the talk, you mentioned or showed that that PRN5 only interacts with PRC2 under cold conditions, right?
Yes.
So I was wondering, I had a couple of questions about it, but I was wondering mostly, do you know if PRN5 is already at the target locus and just it isn't bringing PRC2 in yet, or is also PRN5 coming to the locus dependent on?
Yeah, so we need to differentiate between 2 accession in plants. So when the FLC is actually transcribed, WRN5 is not going to the locus. So WRN5 only goes to the locus in the cold where you have full PRC2 formed. So it doesn't go there before cold, it goes there in the cold, but it then stays there after the cold. So it requires cold to change something that allows WRN5 to bind the locus, and it stays there even after cold. So WRN5 can also have seen the cold.
Yeah. And do you, I don't think you mentioned not knowing yet why the interaction was induced, but since you mentioned the PhD finger was involved, do you think there's any methylation mark on PRC2 that could be triggering this, or is that very wild imagination?
No, we actually thought about that because, of course, of the methylation on Jared2 as well. We thought about this. Because the PhD domain is a non-canonical PhD domain, it doesn't look like normal methylation. We think it recognizes other stuff. So it's likely that it recognizes another protein modification. But we do actually think that what actually happens is that you need to stabilize the scaffolding protein BRN2, and Win3 or cold is probably stabilizing the scaffolding, so SUSI12 in the cold, and that's why you don't have complex formation in the warm, because you haven't yet stabilized WRN2. So there's something in the cold that stabilizes the scaffold, and that's now allow WRN5 to bind. So that's what we think happens in the cold.
Okay. I think Ben had a question, and he can unmute himself.
Yeah, Matthias, great talk. That was really interesting. I was wondering, I had two questions actually. First, I just love the system. I didn't know much about it before. What is the temperature swing when you go from warm to cold?
Yeah, so that's actually, the lab recently published a paper on that. So normally people in our field do this experiment at 4 degrees. So they take the plants from 20 and go to 4. But mineralization really happens at everything below 14. So as soon as you go below 14, you start accumulating Win3. And yeah, there's a whole upstream signaling of how cold is sensed and put into Win3 protein levels. But as long as you're below 14 degrees, you start accumulating Win3.
Fascinating. And then the other question I had was for your nucleic acid binding. You showed that it binds to DNA, and you said it wasn't necessarily sequence specific. Yeah. Have you looked to see if it binds to RNA as well, or just DNA?
Yeah. So we really wanted to bind RNA because I didn't talk a lot about the art, but the lab really focused on non-coding RNA as well. So one idea was that the non-coding RNA was induced in the cold, is what brings in PRC2. So we also tested the binding to RNA, and it does bind RNA as well. We have a slight hint that it prefers some kind of structured RNA compared to linear RNA. Well, that's really ongoing work, but yeah, it definitely also binds to RNA nucleic acid.
Awesome, thanks.
Okay, then as a final question, so Alan Underhill raised his hand, so I will now allow him to ask his question.
Yeah, great stuff. I'm curious is that pattern you see with Win3 GFP temperature dependent in terms of forming those foci? And secondly, do you know anything about the dynamics of the protein within those foci?
So we are just beginning to look at that. So Win3 is temperature depending, so we can only look in the cold, so to say. So as soon as we go to the warm, Win3 is degraded.
Okay.
But I mean, we haven't looked at fluctuating. So as I said, below 14 degrees, Win3 is expressed. So there's a difference between our 4 degrees constant, and if you look at 8 degrees, I don't know. It's possible. But yeah, we are really starting to look in collaboration with some people at the University of York into the dynamics and of these foci.
Thanks very much.
Cool. Okay. I think we'll wrap up there. If people think of other questions to ask later, you could join us after the second talk for the coffee chat, and you can ask them then. Okay, thank you, Matthias.